Okay, now I just want to talk a little bit about mutations. All of the information we've talked about before, when we talked about the specific sequences of the base pairing, and how if you alter even just one of those nucleic acids, if you alter just one of those binding sites, it can lead to a change. So anything that alters the base sequence of the DNA leads to a change. That change is referred to as a mutation. You certainly should know that definition. Most common mutation in base substitution or point mutation. This is where a single base in a DNA is replaced by a different one. Such a substitution is likely to result in the incorporation of an incorrect amino acid in a synthesized protein, which will result in what's referred to as a missense mutation. Such an error creates a stop codon which stops protein synthesis, which is not good if protein synthesis involves the utilization, say, of an energy source such as glucose. But this resulting in a nonsense mutation. Deletions of additions of base pairs result in what's called a frame shift mutation. So we have point mutation that you should be familiar with the definition. We have nonsense mutation, which you should be familiar with that definition, and then frame shift mutation, again, where you should be familiar with, and then the missense mutation as well. So you should be familiar with those definitions. Spontaneous mutations occur without any known intervention, mutation-causing agent. Many chemicals and/or radiation exposures bring about these types of mutations. Chemicals and/or radiation is referred to as mutagens, substances that can bring about mutations. There are chemical mutagens that we hear about in the environment. Some include nitrous acid, which is a base pair mutagen, causes adenine to pair with cytosine instead of thymine, which is a no-no. Other mutagens are nucleoside analogs. In other words, they give you the appearance. They fool the system. They make the system think that the correct base pairing has occurred, but it's actually an analog, a nucleoside that looks similar to but not the same as the appropriate base. And basically what happens, because it's structurally similar, it will get incorporated and create a DNA error. Now, this sometimes is done intentionally, especially if you're trying to destroy particular microorganisms, or if you're attempting, when we look at a biological system, perhaps there's a particular type of pest, either an insect or animal, that we want to destroy. You can actually apply a mutagen, manipulate the base sequences, and causing a DNA error in a particular, especially in a specific protein, say a protein or an enzyme that's involved in the catabolism of glucose. So now this organism that's glucose dependent, like humans, we're glucose dependent, if you create a mutation where we can't produce the enzyme that breaks down glucose for energy utilization, say one of the enzymes in the Krebs cycle or the TCA cycle, we alter protein synthesis, we can no longer carry out the Krebs cycle, we die. You can do the same thing for microorganisms. You can do the same thing for pest control or insects. We see this genetic manipulation being used on a lot of agribusinesses now, utilizing particular mutations that only cause harm to the particular pest that you're trying to get rid of. So it's very important. Examples again of agents that may be used, 2-aminopurine and fibromouracil. These are analogs to adenine and thiamine. Some antiviral drugs utilize these nucleosides as analogs, and again, it will inhibit the growth of particular viruses. Very common in HIV therapy to manipulate or inhibit particular enzymes that virus is dependent upon to be able to carry out its virulence. Again, by manipulating, by inducing an analog, which is basically a mutagen, you actually interrupt the protein synthesis of that particular microorganism, and thus you can kill it or inhibit its growth. Frame shift mutagens, examples include carcinogens, benzpyrene, which is found in smoke and or soot. You ever see the commercial with the chimney sweepers? And they're talking about they got like a new way to get the product around, and they're coughing and the smoke comes out. Anybody ever see a commercial? The guys laugh and the smoke comes out. Nobody's seen that commercial? I think that's a great commercial. Anyway, they've been ingesting benzpyrene, which the chimney sweeps. They did a lot of research studies in Britain where chimney sweeps are big business, and these guys would go in chimneys and they clean out chimneys. They come just covered with soot and stuff, and you inhale all that stuff in the lungs. And it's a mutagen. It leads to cancer. Still hadn't nobody seen that commercial. That's painful. Okay, found in smoke and in soot. Aflatoxin, certain types of mold, toxic mold can produce aflatoxins. And then there are particular types of diet-erican dyes that are produced as well. These cause frame shift mutations in biological systems. And then radiation, important to know about radiation. Different types of radiation. There are ionizing radiation. There's ultraviolet radiation, which is considered to be non-ionizing radiation. You need to know those definitions. Ionizing radiation, such as X-rays, gamma rays, mutagens, they can damage DNA. Ultraviolet radiation, ultraviolet light, non-ionizing radiation, another mutagen that affects DNA. Certain light repair enzymes can repair ultraviolet damage in a process stimulated by visible light. Nucleotide excision pairs use enzymes to cut out distortions of DNA and synthesize replacements. Now, again, just as a review, mutations, change in genetic material. Mutations can be neutral, beneficial, or harmful, depending on what we're trying to manipulate and what we're trying to control. Mutagens are basically agents that cause mutations. Spontaneous mutations occur in the absence of mutagens. Base substitutions and or point mutations, changes in one base. Missense mutation results in the change of amino acids. It's a little illustration for you demonstrating those. Nonsense mutations result in nonsense codons. Then frame shift mutations are basically the insertion of or the deletion of one or more nucleotide base pair. Ionizing radiation, these are X-rays, gamma rays, cause formation of ions that can react to nucleotides. Nucleotide excision pairs repairs these mutations that have occurred. Now this has a point of limitation as well. Extensive mutations in base pairing, not even the nucleotide excision can repair these multiple mutations that can occur. UV radiation can cause a thiamine dimer, basically just a change in exposure to ultraviolet light, causes adjacent thiamines to become cross-linked, become thiamine dimers, and thus interrupts the normal base pairing. The mutation rate, the frequency of mutations that we see in the environment. Mutation rate is a probability of gene mutation each time a cell divides. DNA replication is a very faithful, and again, there's a sequence of steps to help prevent mutations from occurring. So DNA replication is very faithful. Only about once in a billion base pairs do we see replication errors occur. Mutagens basically increase the rate of such errors to 10 to 1,000 times in frequency. Spontaneous mutations occurs 1 in 10 to the 9th power. Replicated base pairs, 1 in 10 to the 6th power of replicated genes. Mutagens increase from, as you can see, 10 to the -5, 10 to the -3 per replicated gene. So the frequency is few and far between. You don't have to memorize those numbers. That's not why I'm putting them up there. It's just to give you some idea of how reliable that sequence is. But what affects the reliability? Well, exposures to various types of mutagens. If you're a chimney sweep and you get exposed to benzpyrene a lot, then it certainly increases the probability of a spontaneous mutation. If you get exposed to other carcinogens in our environment, as we know in the biological system, there are a number of other agents that we can get exposed to. But when we talk about microorganisms, there are times in genetic manipulation that we actually will engineer particular microorganisms to cause these types of mutations to occur intentionally. Mutants basically which occur at a low rate, can be identified more easily with bacteria because bacteria produces very large populations quickly. Positive or direct selection is illustrated by plating out bacteria in a median, say, containing penicillin. Mutations which have occurred, if there's a survivor that is in a penicillin type of median, then we know that these are penicillin resistant mutants have been isolated. Now in a hospital setting, this is very important because we need to know what microorganisms have mutated and are now resistant to various types of drug therapy that we select. And again, we do this by using a selection medium or selective medium. We already talked about those in the previous part of the course. But again, survivors in a selective medium, you would obviously know that these organisms have now mutated and they're resistant to penicillin. And it's very important in hospital settings to be able to identify what microorganisms have developed these types of resistance. And it's not just for penicillins. Other antibiotics, cephalosporins, or macrolides, all of these types of antibiotics that we use, they're not effective on certain organisms because they've mutated, and it's important to know. We may have to change antibiotic therapy to, say, an aminoglycoside, which is a different type of antibiotic, differs from penicillin, differs from the macrolides. And again, you may have to utilize that type of drug therapy to be able to kill these microorganisms. What we really find out, though, is how quickly microorganisms can mutate, especially in hospital settings. And so it's important to be able to culture out selectively these microorganisms to determine whether or not they have antibiotic sensitivity or not. Nutritional mutants, auxotropes, these are unable to synthesize nutritional requirements such as an amino acid, an activity that the parent type was potentially capable of doing, but now we see the offspring may or may not be able to do so. If you want to isolate one of these particular types of organisms, colonies growing in, say, a master plate containing a complete median can then be transferred to a replica plating. This is an example of indirect selection, whereas before we preferentially put it in a selected median. So again, just a different type of mechanism that you can use to be able to isolate or identify particular types of microorganisms. In this particular case, it's based upon what the nutritional requirements are for those types of organisms. In indirect selection, basically the process, and again, remember, I'm not going to ask you a lot about processing because I don't expect you to be in a lab doing these types of procedures. So just generally, I want you to appreciate the concept of indirect selection. I'm not going to ask you, unlike the gram stain, which is very important, unlike the gram stain, I'm not going to ask you to walk through the steps of indirect selection. But basically, in a lab, what you would do is you use a sterile velvet pad pressed onto a master plate. Colonies would be transferred simultaneously to a minimal medium which lacks the essential nutrients for such of an organism to grow. Then the auxotrophic mutant will fail to appear on the minimal medium. This is indirect, or a form of indirect selection. So another study referred to as the Ames test is based on the ability of a mutated cell to mutate again and to revert to its original form. You do need to know that definition. The organism that's used in this model is Salmonella. Salmonella, which has lost the ability to synthesize the amino acid histidine. It's plated out on a minimal medium without histidine. In the Ames test, we see that the test chemical basically is placed on the plate. A mutation of Salmonella to the normal histidine synthesizing, remember before we talked about lactose sensitizing, but now we're talking about histidine synthesizing forms, are indicated by colonies growing near the test chemical. High mutation rates are based on characteristics, effects of carcinogens, and so this test can be used to help determine if the chemical is mutagenic or possibly carcinogenic. Okay, need to understand that. That's the principal purpose behind doing the Ames test. And again, the model that's used there is Salmonella. Mutation of Salmonella to normal histidine synthesizing form. Thus, the test will help to determine if a chemical is mutagenic or possibly carcinogenic. Now, there are some limitations on the test. It cannot determine the degree at which a chemical substance is mutagenic. In other words, what we test on in a microorganism, and for that matter, what we test on in a lab rat, may not necessarily be applicable to the human system or another biological system. Remember, a mouse is different from a rat, and the enzymes secreted in proteins secreted, say, in the rat's liver is different from what's secreted in the mouse's liver. So a substance that may be carcinogenic in a mouse may not be carcinogenic in a rat. That's proven. So take that same analogy and try to apply what's the enzymes that are produced in the liver of a mouse compared to the liver of a human. You cannot extrapolate the information that's gained in that laboratory study and apply that in every case to the human system. I mention this to you in microbiology because there's a lot of litigation out there these days. And litigation quickly says, okay, we took this chemical substance, it caused cancer in 75 of 100 rats. Okay, that means it causes cancer in humans. Okay, well, no, it doesn't. That's not what that means. It means that it causes cancer in that rat at that dose. But at a different dose, would it cause cancer in that rat? And at a different dose in humans, it probably doesn't cause cancer at all. So you cannot always use this information. And so the limitation is, cannot determine the degree in which a chemical substance is mutagenic. There has to be a specific dose. We have to be talking about specific enzymes or protein synthesis that occurs in a particular organism. You cannot always take that given information that you do in a laboratory study, especially if you do it on something other than a human, and try to use that information to present or to prove disease somewhere else. And that's true all the way. It's not even accepted in court to be able to use that process anymore. There's a law called Daubert law where you have to be able to prove this was done on human studies. It involved the same process. There has to be a specific dose for a mutagen to cause a problem. It has to have a specific target organ as well for the mutagen to be able to cause a particular problem. Selections. Positive and direct selection detects mutant cells because the growth appears to be different. This is a review. Negative indirect selection detects mutant cells because they do not grow. And just some replating techniques of the Ames test. Again, you don't have to know that process, just need to know the general principle behind the Ames test. Salmonella infections. Salmonella infections remains of great importance in the poultry industry. Now, I happen to be from a poultry state originally. Everybody had chicken farms. I used to catch chickens, so salmonella was a big disease there. And now in the poultry industry, because it's now in the forefront of most of the concerns of those who produce salmonella, salmonella infections are of significant importance also in the household because many of us will bring raw chicken products into the household for preparation for cooking. Salmonella infections remain great importance in the poultry industry, partly through the direct commercial loss of bird infections, but more specifically important through public health concerns. There's the pyramid or pyramidal structure that's been set up in the industry, which means that infections higher up on the food chain may be magnified at the food producing end of the chain as well. Currently, the most important aspect of this problem seems to be the persistent infections by breeders at the farm level, special attention to disinfection, house designs, vermin control, levels of greatly reduced contamination of birds destined for human consumption. It's a cornerstone of breaking the cycle of salmonella infection and contamination. During the recent hurricane, I'm from Mississippi originally, and during the recent hurricane, Mississippi is known for two things: pine trees and chicken farms. And many of the chicken farms were destroyed because they're, I don't know if you've ever seen a chicken. Anybody ever seen a chicken house? You know a chicken house? Okay, chicken house. They're long structures. They're not very sturdy. I mean, they're sturdy enough to grow chickens, but they wouldn't withstand the force of a hurricane. So a tremendous number of chicken farms were killed, and the chickens were killed And got to remember, not only were the chickens killed, but then the groundwater gets contaminated from the bodies of the chickens because it's still raining during the hurricane. So there is this fear that there's going to be this huge salmonella outbreak in the state of Mississippi, probably Louisiana as well. Now, public health really hasn't found that to be true. There's a lot of testing going on, not above the level, the acceptable level of salmonella contamination that's already in the environment. But there's this big fear that salmonella outbreaks are going to occur because it's related heavily to the chicken industry. Salmonellosis is basically a disease caused when animals and people become ill from infections by salmonella bacteria. All domestic and wild animals are at risk of contracting salmonellosis. Disease also spreads easily from animal to human. Usually affects the intestinal tract, causes severe diarrhea, often ending up in the death of animals. Can cause severe diarrhea, abdominal pain, and cramping in humans as well. Salmonellosis caused by a specific salmonella strain, the Dublin strain in calves, often confused with pneumonia. There are other species of salmonella that can cause abortion in cattle as well. Notable feature of salmonella bacteria is that it will invade birds and animals which have become carriers of the disease. Carrier animals appear normal or to have recovered from the disease, but they shed the bacteria into the surrounding environment through manure, saliva, or other discharges. Carrier animals cannot be cured by treatment with antibiotics or other drugs. Organism that's found in the intestines of virtually all animals, including poultry, reptiles, livestock, rodents, birds, and humans. Animal-to-animal spread and salmonella-containing animal feed maintain the reservoir. The most common sources for humans, and you need to know this, for salmonella, most common sources: poultry, eggs, dairy products, foods prepared on contaminated work surfaces. So you take the chicken, you put the chicken on the counter, you chop the chicken up, you feed it to your pet lion. But then when you prepare your food on the same counter, you contaminate your food with the salmonella that was on the counter. So again, that's why food preparers, especially restaurants and whatnot, have a certain level of standard that have to be maintained for surfaces to be cleaned. Also, temperature. This organism is temperature dependent, so obviously cooking the food can change, can kill the bacteria, would make it safe for ingestion to some degree. Our main defense against the disease salmonella is the stomach acid. Salmonella is very sensitive and it will die in the presence of stomach acid. However, that being said, remember the bacterial load. Depending on the bacterial load and the transit time through your stomach, perhaps all of the bacteria didn't get exposed to a high enough level of the gastric acid that's in your stomach and passes into your gastrointestinal tract, into the colon or small bowel, before it has a chance to be able to be killed by the digestive enzymes. Now, another concept that I want you to appreciate is genetic transfer or genetic recombination. Genetic recombination, you need to know this definition, is the arrangement of genes to form new combinations. If two chromosomes break and are rejoined in such a way that it becomes or is similar to a gene, it's reshuffled between two chromosomes. This process is referred to as crossing over, form of recombination. The original chromosome, each have been recombined, so now they carry a portion of the gene to the other chromosome. Recombination, more likely to be beneficial than mutation, and also occur in several ways. In all cases, we see the donor cell gives a portion of the donor DNA to different recipients. The recipient is basically called the recombinant. The DNA from the donor is added to its own DNA. Now, you can have what's referred to as vertical gene transfer. Need to be familiar with this. Vertical gene transfer, it occurs when genes are passed from an organism to an offspring. Vertical transfer. Bacteria can also pass laterally to other microbes in the same generation, known as horizontal gene transfer. Again, from parent to offspring, from one organism to the other. Horizontal gene transfer, vertical gene transfer. Vertical gene transfer occurs during the reproduction phase. That makes sense because it's being passed to the offspring. Horizontal gene transfer, the transfer of genes between cells of the same generation. Transformation in bacteria. DNA in a solution is transferred from one bacterial cell to another. This can happen. Again, this is in bacteria now. It's not in human systems. Process occurs. Can't put you all in a vat and you change genes like that. Doesn't happen. The process occurs naturally among very few genera of bacteria. Usually occurs when a donor and a recipient are closely related. It usually occurs in the log phase of growth. Takes of DNA fragments. The recipient cell must be competent. That is, the cell wall of these organisms must be permeable to large fragments of DNA molecule. Now, remember, in the human system, when we look at the nucleus, the nucleolus, and then we look at the nuclear membrane, remember, the portions of DNA are too large to be able to transfer like that. That's why we have to have messenger RNA, smaller fragments that are able to pass through these nuclear pores. But you can see here in these types of microorganisms, bacteria, they can take up fragments of DNA, so the recipient cell, for it to be competent, the cell wall has to be permeable to these large fragments of DNA molecules. So here you see DNA just floating around everywhere, but we don't see that in our system. And again, these are some of the steps in the transformation, the recombination process that can be manipulated in the laboratory animal. There is a process referred to as conjugation, which requires direct contact between living cells and opposite mating cells. I'm not going to go into the particulars of conjugation of bacteria, so you're not going to have to know the F site. It just basically, in conjugation, it does require direct contact of the living cells of these opposite mating units to be able to take place. Now, in your laboratory study for this week, I believe you did plasmids, right? Plasmids. So you know what plasmids are. They're types of plasmid. These are free-floating genetic elements in cells, important in being able to use, and plasmids are used a lot in DNA recombination. The F factor basically is bacterial chromosomes, which it's not passed, it's not a recombinant, are being produced, but again, you don't have to worry about the F factor at all for the purposes of the course or the examination. In genetic recombination, just appreciate this process. You see an exchange of genetic information, or exchange of genes between two DNA molecules. Crossing over occurs when two chromosomes break and rejoin. In the transduction in bacteria, transduction, the phage portion or the phage portion, short for bacteriophage, which is a bacterial virus. A bacterial virus, it attaches to the bacterial cell wall and injects DNA into the bacterium. Normally this directs the synthesis of new viruses. And remember, viruses, I don't know if you, the HIV, was that on this lab too? Have you done the HIV yet? No? Nobody's nodding, so maybe that's on the next lab. But you'll see the process of, I'll get that posted up, because it's a nice little film to look at that shows you how a virus attacks the human cell, and how not only does it attach to the human cell, but then it obtains an intracellular location, and then it obtains an intranuclear location. And viruses are tough organisms, because not only do they enter into the cell and get an intracellular location, they get into an intranuclear location and then utilize the material, the genetic material inside the nucleus to carry on their own replication. So as we see here in generalized transduction, we see the bacteriophage, or bacteriophage, however you want to pronounce that. Basically, a bacterial virus attaches to the bacterial cell wall, injects the DNA into the bacterium. Normally this directs the synthesis of new viruses. Sometimes, though, we'll see bits of bacterial chromosomes that are accidentally incorporated into a viral DNA. When these viruses infect a new host cell, they can incorporate this genetic information from previous bacterial hosts into new bacterial hosts. Specialized transduction, only certain bacterial genes will be transferred in that process. And this illustration, again, out of your book, just shows you basically, and again, I'll post up, if it's not already up there, I'll post, there's a media presentation about the HIV virus, and basically it shows how the virus can attach to the bacterial coat, or in that particular case, to the human cell, and then will obtain an intracellular location, just as we see here with the phage protein. And then the phage DNA and the proteins are basically made because it can obtain, develops a quick relationship with the genetic material in the particular microorganism, in this case, that we're talking about. And then replication will take place. In the case of bacteriophages, you can see that during this assembly process, how the incorporation of genetic material along with the bacteria, the virus and the bacteria, are incorporated into the same cell. And then a recombinant is produced at the other end, with genotypes is different from both the donor and the recipient, but contains components from either. Another structure, plasmids, basically are circular pieces of DNA, replicate independently from cells or chromosomes, usually carries only genes that are not essential for the growth of the cell. Dissimulation plasmids basically code for enzymes that are utilized for unusual sugars and hydrocarbons. Plasmids cause a synthesis of bactericidin, which are basically toxic proteins that kill other bacteria. There are resistant factors that carries genes that are resistant to particular antibiotics and other antimicrobial factors. Some R factors contain two groups of genes, resistant transfer factors, coding for plasmid replication, R determinants, and basically coding for enzymes that inactivate antimicrobials. So should be familiar with the term R factor. It's just a simple definition to be able to recognize, again, the genes that are responsible for developing antibiotic resistant. And in a clinical setting, again, this is important. You'll see many microorganisms that will develop these resistant factors. Again, these are different types of plasmids, conjugative plasmids, which basically carry genes of sex pili that transfer the plasmid. Dissimulation plasmids, which encode for specific enzymes for catabolism, for unusual compounds, specific types of sugars that may be used as an energy source for a particular type of organism. And then R factor, again, is the antibiotic, or encodes for the antibiotic resistance. This is an illustration out of your book that not only an illustration but electron microscopy as well of plasmas, these circular protein structures. Transposons are basically small segments of DNA, small segments of DNA that can move. They basically are transposed from one region of the chromosome to another. They basically sometimes are referred to as jumping genes. This movement is relatively uncommon, but would basically be the same as spontaneous mutation rates in certain bacteria. Certain types of transposons, the simplest transposon is basically called an insertion sequence. They code usually for an enzyme. The enzyme referred to obviously as transposases. They cut or ligate DNA into recognition sites, and at the recognition sites, these are short regions in the DNA where enzymes will recognize a particular recombination site of the transposon. If you see a particular sequences of the bases, they code for recognition. So again, you can see the importance of these base sequences being correct or complementary. You can have complex transposons which carry other genes, such as those conferring antibiotic resistance, as I've talked about previously. Transposons are a very powerful mechanism of movement of genes or genetic information from one chromosome to another and between species. Transposons. Just another definition for you to be able to appreciate this illustration out of your book again. Segments of DNA that can move from one region of a DNA to another, contain an insertion sequence of cutting and or resealing the DNA. These all will lead to a mutation, an organism that's now going to be different from what the parent organism is. These complex transposons